Hello, everyone, and thank you for joining us today for another ACE trending paper presentation.
Hello, everyone, and thank you for joining us today for another ACE trending paper presentation. Hello, everyone, and thank you for joining us today for another ACE trending paper presentation. Hello, everyone, and thank you for
who will present a recent Nature paper titled Deciphering Interaction Fingerprints from Protein Molecular Surfaces Using Geometric Deep Learning. With that, and without further ado, I pass the mic to Florian. Florian?
Okay. Hello, everybody. Thanks for introducing me. Gonna start any second now. So I'm gonna welcome everybody to my talk. So we're gonna talk about a recent paper about predicting protein like surface features attributes using some state-of-the-art geometric deep learning method. So first of all, a little bit about myself. So I got my computer science degree in bioinformatics from University of Munich and did a postdoc at the University of Toronto. And in total, I worked for roughly 10 years in the machine learning field, 6 of which were in bioinformatics. And after my academic career, I worked for a marketing company doing machine learning on big data. And currently, I'm working at BMO Capital Markets doing various machine learning solutions and for capital markets problems. And a little bit about overview of what my presentation is going to be. So this is going to go through the paper and the major sections of the paper. As you can think, it's going to be like a bit of introduction, talk about the problem and what are we doing, find out of how we're doing it, and we finish up by talking the results and the possible application of the methodology presented here, and in the end we'll have a few discussion points about it. So in short, what they proposed here is a method that takes some kind of surface features. So you give in input a protein, right? And in short, it's going to do some kind of machine learning on the protein surface to extract like the characteristics or the fingerprint of the surface, and then does some kind of convolution on the surface and creates some kind of general hidden state fingerprint description of it. And finally, they're going to show 3 use cases where they can use these kind of machine learning generated fingerprints to do very useful things with it. So to understand what's happening here, we need to talk about what are proteins, what are protein surfaces or surface features, and we're going to have to do a quick crash course into geometric deep learning. So what is a protein, right? So most people, they know they drink protein shakes and then they think, oh, there's some protein in. But when I say proteins, I'm talking about quite something different. So you can think of proteins at the molecular building blocks of your cell. You can think of it a little bit like Legos of your cell. And each protein fulfills a certain function in your cell. And all these proteins, they work together to coordinate and to fulfill all the processes of your cell needs to do. And your cell needs to do all kinds of things. It needs to generate energy. It needs to process some substrate, some metabolite. So say you have a liver cell, it needs to help to do whatever your liver do, or you have a heart cell, it's mostly a muscle cell. So the muscle cell needs to contract and move the muscle. And in general, like the human genome contains 20,000 protein coding genes. And in general, you can think one gene is one protein, but rather you have to think about one gene as one group of proteins. Because you take one gene, and you can make like different mix-ups, remixes of the same protein. Like you take some part out, you leave some part in. So roughly on average, you can up to have 100 different protein products from a single gene. So the total human proteome, and with proteome I mean like all the possible proteins that you see in your cell, is a bit unknown how big it is, but it's going to be very, very big, easily more than 200K, or more than 100K. And to make things extra tricky is that not in every cell all the proteins are expressed. So some cell, like your heart cell and your brain cell and your liver cell and your lung cell, they're very, very different types of tissue. And to achieve this characteristic that you have different types of tissues, different types of proteins need to be present or expressed in that cell. So in general, there are 7,367 genes. So genes that might contain more than one type of protein, like an assembly of protein, and these are expressed in all tissues. These types of genes are called housekeeping genes. They are essential for every cell, so they kind of regulate the fundamental functions of every cell, like things every cell needs to be able to do, like process sugar, or process energy, or do some signaling. So if you have a mutation in these cells, like a non-neutral mutation, like a mutation that does cause some deficit, then it's going to have a great effect on your body. So how are proteins made? So if you have spent some time or attention in biology class, I think you should know that every cell contains DNA. So this is DNA. You can think of the blueprints of all your proteins. And in the process called transcription, the RNA polymerase translates DNA to a kind of pre-mRNA. And this pre-mRNA is processed. Some pieces of the pre-mRNA are spliced out and modified. And this creates mRNA. And the M stands for messenger. So this messenger RNA is, in a nutshell, the sequence or the blueprint of the protein. So this sequence of the protein gets translated to peptides, amino acids. So in your body, we have 21 essential amino acids in your body, and proteins are built out of these amino acids. So you can think of a protein as a long sequence of these amino acids. And from the mRNA, essentially contains the information what sequence of amino acids you need to build, given from the transcript. And on average, a protein is, if I remember correctly, 100 to 200 amino acids long. And this primary sequence of amino acids, say, you have alanine, glutamine, whatever, is called the primary structure of the protein, right? So this is just, think of it as a long word, right? And just all those letters back to back to back. Just a long sequence of words. And what is happening is that these sequences of words, they kind of organize themselves in something we call the secondary structure. And there are three types of secondary structures. One is called the alpha helix. It's kind of like a helix shape. And then the secondary structure is like beta fold bloods. These are kind of like beta sheets. And I have a 3D structure I'm going to show you later on to further show you what I'm talking about. And loops. And loops are kind of like free, unorganized sequences in your protein. And when all these secondary structures, they fold together, and this is a very complicated problem to solve, just to go from the primary structure of the sequence of the protein to the 3D structure of the proteins, is already a very difficult problem. And last year, there was a breakthrough paper, it's called Deep Fold, AlphaFold, which is also something we can talk about one time, where they use deep learning to predict the protein structure, giving the primary, like the sequence of the protein. And what you can think of is that the alpha helixes and the beta sheets, they're very stiff. They can move a little bit, but they just need to stay information. And those loops are like movable parts in the protein that kind of connects these secondary structures. And last is the quaternary structure. So what is the quaternary structure? This is when two or more proteins interact with other and form a protein complex. So every function in your body are mediated by these protein complexes. These are multiple proteins binding together, forming a stable protein complex. And these protein complexes, they have some type of function. they have some pocket or some area where some substrate can bind, and then some chemical reaction happens, and then you have the products of that substrate or the products of that chemical reaction. So just to give you an example, so recently we're talking a lot about coronavirus. So let me go here, and this is the COVID-19 main protease and complex with an inhibitor N3. So they have an inhibitor N3 here, the small compound, and this kind of inhibits the protease of the COVID-19, and this is the 3D structure of the protein. So if I zoom in here, so when you can take a look here, you see this kind of spiral shaped structures, right? So these are the alpha helixes, and then between the alpha helixes you sometimes see like this kind of thin ribbon, this kind of unstructured thin ribbon. These would be loops, and the other alternative would be the, here you see this kind of arrows. These are would be the better sheets. They're kind of hidden behind the alpha helixes, but you cannot see it. And this shows you kind of the 3D structure of the protein. And this is called like the biological assembly, because this is like two types of the same protein, like some two asymmetric units binding together to form the protein complex. So these are two identical proteins that bind together and they form the protein complex. And this is what something happened rather frequently in biology, because the way you have to think so, if the mRNA gets expressed at a certain location in your cell, and then there's most likely multiple copies of the mRNA expressed at the same time, creating multiple these kind of single individual units, and they're a co-location. And this is a good opportunity for them to find and bind together doing a multi-protein complex. So as you see, this would be the tertiary structure of the protein. And now in the biological assembly, you can think of it a bit like the quaternary structure. And to give you an idea about the surface, so this kind of shows you the surface. If I'm if I'm correct, it also shows you like the electrostatic charge on the surface, and this kind of shows you the surface of the protein complex, what it look like. So a lot of the machine learning we're gonna talk a lot about surfaces, and you kind of think about the protein as like a 3D object, right? And a 3D object can be any kind of object; doesn't necessarily need to be a protein. And let me quit my email. And this will have some kind of all kinds of surfaces on top of it. And now they want to do what it was. The current state of the art was people do some kind of handcrafted features that describe certain patches or areas on the surface. And then people say, okay, now we are living in the deep learning area of the whole thing. And instead of doing all this kind of handcrafted, why not just use some kind of machine learning algorithm or application to help us generate these kind of features for the surface? Well, I cannot see the Slack chat, by the way, when I'm presenting. So if there's something, just let me know. And as I just showed, like sometimes you have this kind of electrostatic surfaces. This shows the electric charge on the surface, or like the accessibility of water to the surface. And sometimes on or every protein, like some areas in the protein are special. These are the active sites. These are the sites where the magic happens, right? This is like some kind of pocket in the protein where a substrate can bind, and then or a compound can bind, and it can either block the active site or it can be help the protein to do whatever the protein needs to do. The fundamental way of describing this is called the key and lock system, right? So you say you have some enzyme and substrate, and they enter the active site of the protein, and once they're both bound together, the protein complex or the protein can do its molecular function. It conducts its molecular function, and most of these times and NADP is burned. This is kind of the energy, the fuel of the cell, the fuel of your body. So if you weigh 80 kilogram, roughly daily, you're going to produce 80 kilogram of this compound, of this energy compound. And once it's finished, this is going to be released, the product is going to be released, and the active site is free again, and another substrate enzyme can bound to the active site, and it can perform the action again. So that's how this would conclude the part about the proteins. So we're going to talk more now about what happened in the paper, or what they've done in the paper. So if there are any questions about this kind of background stuff, feel free to post it or to ask. Else I'll just continue. So here in this paper, the main part of the feature generation is generating these patches and the features for the patches. So what they did is they used some special software to determine or to create the molecular surface of the protein, and then to make sure they have like a uniform size, or some, just to keep it consistent, they downsample the mesh size to 1. So think about, in the lower corner you see like a protein, and then mesh, they put this kind of node vertex lattice area on top of the surface of the protein. And they create like these patches for each surface. So what they do is they take a node, like any of the node of the mesh, or every node of the mesh, and then they do a 9 or 12 angstrom like circle on top, like geodesic distance on top of the patch. So if it's like weirdly shaped, you have to take this into account. And for each mesh node in that patch would be in neighborhood of the center node of the patch. So you have here this black dot on the left side in a green circle. Sorry. You have the black dot, and then the red line would be the radius, and then this would define kind of the patch. And all the mesh nodes, vertices in that green area would be inside this patch. And then they calculate 5 features for each kind of mesh node inside this patch. One is like something to describe the shape, is like a U-shaped or whatever. One is to describe the distance of the, like distance-dependent curvature, is like describing the curvature. And they have 3 chemical features. One is like the hydropathy, like how hydrophobic, like is it good, can water access that area, or can water not access that area? And in general, it's important because if you have a very water accessible area, like water soluble area, then it's most likely the surface of the protein. And if it's a water inaccessible area, it tends to be the active site of the protein. One is for the electrostaticity of the surface, and the last feature is just the amount of free electron or protons on the surface of the protein, like the electric charge. So the problem is, now I want to do convolution, right? Or they want to do convolution. We want to have an area, we have a surface, and we want to describe that surface or the relationship of all the nodes in that surface to each other in respect to those features, those 5 features we declare. And the problem is that it's like a 3D surface, right? And say, you cannot just simply do 2D convolution or whatever convolution on it, because it's like not very uniformly shaped, or it does not, you can't take the leverage, the structure of a 2D image as you can do for the 3D surface. So the neural network active feature they use here is from a previous paper they published, and it's called so-called MoNets, that are mixture model networks. And I'm gonna describe a little bit in detail now how this works. And the way it works is that if you want to create the features for a patch, then they just do this kind of convolution for each node in the patch, or for some nodes, or for different node patch locations, like different nodes in the patch. And you can think of it a little bit like sliding across the patch with your convolutions. So the advantage is, say you have 2D convolutions, or if you remember, you have you take a square or a cutout of the bigger image, and you have some kind of filter you apply to the image. And this works very neatly, because if you take a cutout of the image, it's going to be a matrix, right? And all the relationships of all the pixels in the matrix are like fixed from the position inside of this matrix. Say you have a 3x3 matrix, then the center cell would be the center pixel, and the lower left cell would be the lower left pixel of that cutout, right? So the spatial information is preserved by just looking at it as a matrix on it. Unfortunately, it doesn't work that neatly for 3D structures. So for 3D structures, as you can see on the right side, what a lot of people use, or we use here, is kind of like polar coordinates. And the way it works is that you have some kind of geodesic distance. So if you look at the left figure for the surface convolution, it's like such rings, right? Rings around. And these rings around, they kind of determine like how close you are to the center, right? So the red ring is the furthest away, a yellow ring is like a bit closer, and so on. And the second information you use is like your degree, in which degree you're looking, right? So say red, blue, teal, and yellow is like if I would take the 360 degree around that node, and I would split it up into 4 groups, or I would split it up into different like fields. So this is what it would look like on the left. Like you have some, say, 2D, and you see for each point, you say I have a row distance, or r distance, and a row degree that looks in relationship to the center point. And similarly, this is done for the protein surfaces. Just like as the 3D model, we have here 3 patches, the white, the orange one, and the light red one. And these are kind of your angular coordinates. And then it says, okay, in which angular field I am in. The second component would be the distance, or the radius, like how far away are you, like geodesic distance from the center. And the way how MoNet integrates this information into the neural network part is, okay, we have f of y. So what they say is that we have a patch, right? It has a center node and a set of neighbors. And what it does, it kind of averages the features over all the neighbors, giving a distance function, and a Gaussian mixture kernel. And the way the components work, you see, you're going to sum over the neighbors of x, and u x of y. Like I said, u of x of y is this kind of angular and radial coordinate. So it's like 2-dimensional. And wj is something just to add a weight onto it. And we do this j times. We have j kernel. So we say, okay, we do it 20 times. So you get like 20 different weightings of the feature, right? Say you do it 200 times, you get 200 different features. Think of it like as your filters, right? And or in the paper, they use it like to define like the number of these angular and radial groups. So we have, say, they have 12 radial and 18 angular, so you have 12 times 18 filters. So this will kind of give you each of these positions. And this u of x and y is just these 2 coordinate features, and this wj is, you see underneath, it is just what is happening is you just subtract the mean, like the mean value of that position, and you multiply it with the inverse of the sigmoid. So what you do is basically you standard normalize kind of your position features, or you make it part of this Gaussian mixture models. And the output of this weight, of this Wj of u, is 1. So you can think of it as just a rescaling of the feature. So I make an average over all my neighbors, and I weight each neighbor based of its position in that kind of polar grid, in this polar coordinates. So if you look at it, how it's done here in the paper is, you see in the center by D, there's this soft learned, they call it the soft grid. It's learned. And what does it mean? What I mean by learned is that when they initialize the model, what they do, they take the initial coordinates, they look at the patch, and then make initial coordinates for every for every mean value, and they have like a fixed standard deviation value. So what they do is they make a standard deviation matrix and then move like a mean vector, and the mean vector is just the mean value of each of these fields. So you see in the where you see the map features to learn soft grid from, in the D box, there's like this polar candidate. So you see they have like 1, 2, 3, 4 radial features and 18 or 12 angular features. So they have 12 times 18 mu values, which is just the average value of each cell, would be the initial value of the mu vectors, and they just have some fixed standard value for all the covariance matrices. So at the beginning, it's just nothing special, just like a fixed, like the mean value plus a fixed standard deviation. And when you train the model, these values, they just get optimized, right? And then they kind of generate these filters, these n filters, right? You do it n times. The problem is like, if you look at the surface patch, your protein has like some kind of orientation, right? The patch has some kind of random orientation. So you might have even the identical patch that might just be rotated, right? You have here patch 1, and it's not a good example because your hands are not overlapping identical, but say you have 2 times the same patch, they just rotate it. So if you would just consider one fixed like angle, you would get 2 different values, right? Just because the protein itself is fully rotated. So the way they deal with it is they do K rotations. So they do the patch, and then they calculate all the filters, and then they do a rotation, calculate all the filters, do another rotation, calculate all the filters, and they do a rotation, and they do this whatever 18 times. And then they take the max value of all the rotations. So if you look in the box, you see that's written on the left side, K rotations and N filters. So what they do N these convolutions or N filter of convolutions, which assigns these WU. You see these cells, they have like a value that goes from white to blue to red. It kind of is like the importance of that cell. And these are these kind of WU values. And they do this rotations K times and then take the max value. And then you get some kind of fingerprint description for that node of that patch. And then they're going to apply whatever neural networks to these kind of convolutions. So are there any questions?
So we had one question, and they asked whether or not we can use 3D convolutions instead of using this method.
What? Oh, you mean like just have a 3D convolution on? Well, the problem with 3D convolutions is you would just slice out like a cube out of the surface if people say 3D convolutions. So the problem with that is that it doesn't take the distances into consideration, right? So if you just use Euclidean distances on the surface for every other point, it's going to mess up your calculations, right? Think you have like a U-shaped, right? You have a U-shaped surface, and you want to compare this node and this node. So you take the Euclidean distance between these nodes oh, they're actually very close. But then if you take, okay, the geodesic distance, so the distance walk along the surface, it's going to be much longer. So if you just say, okay, I just simply naively take like a 3D convolution on top of the surface, it's not going to work that properly.
Right.
If this is what was the question.
So in this work, they're working with surfaces. If we're even using 3D convolutions, what's going to happen to the inside of the material that you're considering? How would you consider that, even if you want to use 3D convolution? Because you're focused on surface, right?
Yeah.
So basically, the inside of the material becomes useless. Whatever data you have inside it, you just want to focus on the surface.
Yeah. So the inside doesn't matter.
Yeah.
That's the other thing. Yeah, I agree.
So another question was about the structures that you were showing, the secondary structures, and they were asking about the arrow, and they were asking if this is an actual physical structure.
You mean on the PDB?
Yeah, that for the protein, your introduction.
Yeah, it is. These are the beta sheets. So these are the arrows, are the beta sheets, and the twirly things are the alpha helix, and then you have the loops. So these 3 types of secondary structures.
So there was. Okay, it seems like we answered their question, so we can move on to the discussion. We're not going to have a break for this session.
Well, I'm going to talk about the results first.
Oh, yeah, you're going to talk about the results. Yeah, sure. Yeah, go ahead.
Okay, so here this is just 3 case studies. One is for predicting ligands. So you have an active site and you want to know, oh, can these ligands bind to the prediction or they cannot? And the second point is they want to predict protein sites. And the third point is they want to predict protein-protein complex prediction. Like, are these 2 proteins interacting or are these protein not interacting? So for the ligand binding, what they do is say for a given protein surface, predict what ligand they can bind, and they start with 7 ligands. So that's one of the parts I didn't like that much, because the way it is built, you're going to see later, it's going to be some type of softmax across all those 7 ligands. So if you want to predict another side ligand which is not part of those 7, you have to like retrain a model. So maybe a more general approach would be better if it's possible. And so they say it's just more of a proof of concept. So they just evaluate it based on those 7 important ligands. But at the same time, these 7 important ligands are the ones that have most data points available. Most of the data are available. And one part of the data preparation process is, like what they did, they take all the proteins and they cluster them based on sequence. So what you don't want to happen that you have 2 proteins that are basically identical, like virtually identical. They're not identical, but they're virtually identical. And then you split up your data into training and testing. And then you have one in your training and one in your testing. And then you get like extreme good performance. But all you do is just having some like bias, look-ahead bias, like some contamination in your data set. And they say a random model, you have 7 ligands that combined would have like an accuracy of 14%. So this is the general architecture, how it worked. And what they do, they sampled 32 random patches on the protein surface. And then they do this kind of convolution. So they have 12 filters, as you see. So they do 12 filters for each of these convolution. And what they do also, you see, is they do for each feature separate convolution, right? And then they have the 8 rotations. And then they do some ReLU, some max pooling across the rotation. They do some ReLU. And then they do some fully connected layer, and they get like 30 description on this patches, and then they do some matrix multiplication of those features and create the 80 times 80 covariance matrix. And then they flatten this matrix, do 2 layers fully connected, and then a softmax. Sorry. So they're going to evaluate the model. That's considerably better than what they would expect by random. So they see using geometric and chemical features together, they achieve 73% accuracy. Using geometric alone, 55, and chemical alone, 65 respectively. They use this to argue that saying, okay, combining these 2 features has some kind of like co-effect, right? It's better than each individual. So if you combine those, it works better than each individual by itself. And then they did a confusion matrix for all the different compounds. Some of these compounds were harder to classify. Some of these compounds were more difficult to classify. So they see, if you see like HEM is a very big compound, it's very distinct from all the other ones. So they achieve like 94% accuracy identifying it. And similar for ADP is also very distinct from the other ones. However, you have NAP and NAD, and they're a little bit similar to each other. So what happened is going to confuse those 2 together. And on top, NAD, NAP, and SAM, they're all a little bit similar to each other. So they're going to confuse like SAM to be NAD and so on. And the other big error they had was they had a true NADP and then they classified it as FAD. So some of these compounds are more similar to each other. So you put 2 compounds that are very similar to each other and you put them into the classifier, because it's going to struggle more differentiating those 2 than having, let's say, you did 2 similar and some that's very, very dissimilar. So kind of like an out group. It's going to be very easy to differentiate those 2 in groups from that 1 out group. So this leads us to the next test they did, or next proof of concept they did. So they want to predict protein surface. So given a protein surface, predict if a protein or if that surface is interacting or it's non-interacting. And with interacting, is like you have to think about, like I said before, those proteins, they interact with each other, they form protein complexes, and some, they're addressed in the paper. I don't think I showed these results, but some protein complexes, they interact in very strong, stable, permanent interactions. So these interactions are a bit easier to predict, and some of these protein complexes are very, very short-lived, transient interactions. So they're a little bit harder to classify. And here you see what they do is they have multiple convolutional layers, right? The previous one had only 1 layer of convolution. Here they have multiple layer of convolutions, and they have like only 4 times 4 on the polar grid, right, for the weights. So again, all the features come in. You do your convolution with your 12 filters and 4 rotations. You do your maximum over all the rotation. You do your specified linear unit, and then you get your updated features, right, for every node. And then for every node, they do the same convolution layer again, and then create the fingerprint, and then they do a sigmoid, all like 3 fully connected layers. At the end, you have 2 output layers, which is like interact, non-interact, and then they do a sigmoid. Could have had only 1 output layer. Yeah, I guess it must be only 1 output layer at the end, because in the end they have a prediction interface score that goes from 0 to 1. And they say, okay, I have an interface score that goes from 0 to 1. 1 means we're more likely to interact, and 0 is like less likely to interact. They used area under ROC curve to evaluate your performance. And they say, okay, given the cutoff of the score, they say, what are the frequencies of interacting, non-interacting points, right? So if I have a score of very low score, you see you're considerably more likely to have a non-interacting surface point And if your score is like larger than 0.5 or larger than 0.6, you're considerably more likely to be an actual interacting point. And again, they showed that combining geometric and chemical features together yields better performance than using them separately. And at least they take a state-of-the-art model, Spider, and they beat that performance from that model. They outperform it. So the last example or proof of concept they show is like predicting protein-protein interaction. And what they did here is very interesting. So first, what they say, okay, we want to create a vector describing the surface or describing the patch of the protein. And once you have generated these vectors, they say, okay, what can you do with those vectors, right? You can do like a nearest neighbor search for these vectors. You can do some kind of arithmetic on these vectors. And the goal is simply, once you have generated these vectors and you know how to perform the arithmetic on them, you create a database of vectors and you can search or process these vectors very easily, very fast. Think of it a bit like, it sounded a little bit to me like word vectors, like kind of like a embedding of the protein surface. I don't know if I was the only one that felt like it. So the data set was 100,000 pairs of interacting proteins, and they just randomly choose what they do. They take a pair of interacting proteins and then randomly choose a non-interacting pair, like a pair or a site that doesn't interact with neither of those two partners, as kind of like negative set. And when they say negative is they have three, basically the input of three sites. One is like the binding, one is the target, and one is the random. And then they do their embedding, and the goal or the error is like to minimize the fingerprint distance between the two interacting ones, between the binding and the target, and maximize the distance between the target and the random. This was the biggest architecture they had. So as you can see, the input is like the binding and the random, and then the target surfaces. And you see they do all those patches. They do this convolution again. They create these fingerprint patches for the binder and target, and then they had some fully connected that put them all together into 80-dimensional like embedding for those both pairs. And then they just say, okay, you want to maximize the distance between those two vectors. And similar here is like they achieve really high performance for their area under the ROC curve. I'm a bit skeptical because if you get like 0.99% area under the ROC curve, it might be something like maybe your targets were not chosen correctly. But also I had no time to really dig into it or see what was wrong. But whenever I see people performing like perfect, which should be a very complex problem, it's for me a bit of a red flag. But I'll just trust them that what they're doing is correct. So this is like the distance score for interacting patches and non-interacting patches. And you see that for non-interacting patches, considerably higher distances, whereas for interacting patches, complete lower distances. So given these two distributions or this model, they can say achieve 99 area under the curve performance. And again, better performance using all geometric and chemical features together. So are there any questions on the result part?
Yeah, we don't have any questions.
I have. Sorry, is there? Yeah, so I have a question. So, in order to make the model rotational invariant, they had to do these rotations, right?
Yes.
Is there another way of doing it that can still have keep it rotational invariant without augmenting the data, or like they have done? So keep it invariant through the architecture itself or through the features that they use, as opposed to just rotating multiple times?
Yeah, I think this is a good question. So if you look in the MoNet paper, they suggest, for this WU, you can apply whatever function on top of it or in front of it, right? And here they choose to do the rotation, right? So if you can think about any other function that can do the same, that is not rotation, then I think you can just use that function. So it should be possible if you can think of it, right?
Yes.
So I cannot think out of it on the top of my head. Like,
yeah, me neither. But
yeah.
We have another question.
Yeah.
So well, I'm not sure. So someone is asking, building off the rotational invariance question, could you use topology in that area, i.e. both protein surface potential and topology are areas I'm not familiar with? That's a comment from the questioner. So could you use a topology on...
I'm not sure what is meant with topology. So they use this is what this model uses topology, right?
Right. I think the topology of all these patches are similar. So yeah. Okay,
I wouldn't know.
There are these. That's all for the questions for now.
So we have a few discussion points from me and from the facilitators. So one point is like what they did here, they trained like one model, right? So they trained for each case, they trained different models, right? So what I think would be possible is like similar what we see with Inception or other like large convolutional image classifier is just to train like a large classifier, like convolutional protein surface classifier, and then just use transfer learning just to perform similar in all those categories, 3 categories. And I think the advantage is that once you created this big neural network model, other people, if they want to leverage this architecture, they don't have to retrain from scratch. In my opinion. And the other thing is like, the other discussion point is like, or are there any questions, comments to that statement?
So I think what you mentioned is correct. Previously, you mentioned that these look like word embeddings. I feel like that's exactly correct. What they're doing is just a moving window on the protein surface, and they're selecting patches. If you think about it, those patches could be words, and they're basically looking at the whole protein surface as like a sentence. So they're creating word embeddings out of each of them. On the point of whether or not this can be used as transfer learning, because of the fact that we can look at it as word embeddings, I think it can be used as in the case of transfer learning.
But even without word embedding, right? If you think about just the ResNet or whatever image classifier, they also use the transfer learning. They just change they retrain the last 2 of n hidden layers, right?
Yeah. So they usually for transfer learning is you can modify it and change the model to turn it into feature extraction. So I think with this model, it should be possible to do something similar. The issue is getting getting all the resolutions and everything right to feed the data into a model that has already been trained. I think with other transfer learning processes, you have to do the same thing. So I think this point is correct. Karim, what do you think?
Well, I would be concerned about to use this model, transfer learning. The training data needs to be comprehensive enough to cover a large set of what you would need to do in your other applications. So like for word embeddings, if you train on Wikipedia, that's already a very large corpus that covers a lot of the English language already. I'm not an expert in this, but I don't know if the training, the data that they have used is also similarly comprehensive.
The type of data is relatively, I think it's time consuming to process. And I think they have made selection as to how much data they're using. I believe doing something similar to actual word embedding, where you have to train the model for a long time and with enough data, it is possible, but I don't think the current model that they have is going to be good enough for transfer learning. But maybe in the future they can investigate that. That's what I think. Florian?
Yeah, I agree. So my next point would be, so when they trained the ligand predictor, they have those 7 ligands, but these are very important and very abundant ligands, so there's abundant of structure available. So I think if they would train it to predict like different ligands, it would do much worse because there's not that enough data available. And as I said, from the basic how the structure works, right? Like the input was like it was just a softmax, right? It was just a softmax, like supervised learning, right? Mm-hmm. So it would just say, okay, giving this ligand is going to predict one of those 7, right? So even if you want to add another ligand, you have to completely retrain the model. So it's going to be very, very difficult to use it as it is. Say, I want to test like this large amount of, say, 100 compounds or 500 compounds, right? Mm-hmm. So if they would be able to build an architecture where the input would be like a protein and a ligand, right? And then they would take these 2 things into account if it would bind this ligand. But then again, you kind of need to do some kind of surface description of the ligand as well, right? So similar how they do the PPI interaction, you can do like the protein and 2 ligands, right? And one ligand is binding and one ligand is non-binding. Yeah. I think this, if you could build this, would be much better.
Discuss. Yes. No, that's definitely a good point. So in that case, I'm not familiar. How many possible ligands can be there?
Well, you can build artificial ligands, right? You can build artificial compounds. So there are bioinformatics startups, a lot of startups, where they try to predict where they try to use machine learning to create a synthetic and artificial compound to bind to inhibit certain proteins or to activate certain proteins.
I see. So the model they proposed with predicting 7 ligands is extremely restrictive in that sense.
Yeah.
Okay. So Florian, on this point, I'm not really sure how they did the ligand binding experiments. Could you explain that again? Because it seems like that they don't collect a lot of information about the ligand when they're running this test.
They don't collect any information about the ligand when running the test. So what they do is they have like those patches, right? Mm-hmm. And then they have a label for every patch, right? For every patch, for each point, they have a label. I bind A, I bind B, I bind C, I bind D, right? It's like one hot encoded. And then they predict the model, and it predicts like a softmax, and then they do binary cross entropy to calculate the error, right?
So what information about the ligand are they
they're not using any
input about the ligand into the model?
No. Ligand is not the input of the model. This is what I'm suggesting. So if the ligand would be an input on the model, then you can put whatever ligand you want in there, right? You're not really restricted to those 7 ligands, because the model they propose works only for those 7 ligands, right?
Okay.
Well, in theory, you can retrain it for whatever ligands you want to, right?
Yeah.
But then again,
yeah, the first time I read this section, I wasn't sure that how they're doing the binding process with the ligand, but now that you explained it, I understand what the limitations are. Interesting. Thank you so much.
And one other point is like it's.
Oh, go ahead. Sorry.
As like it's limited to proteins where 3D structure is available, right? There are a lot of proteins where you cannot create a 3D structure, right? So you can think of it a bit of like a blind spot, right? You can only create a 3D crystal structure of a protein if the protein crystallizes. But there's a large amount of proteins that don't crystallize. So the whole set of the model cannot work on this set of proteins, right? Technically, like you can use AlphaFold or whatever to predict the 3D structure of a sequence, right? But I'm pretty sure it wouldn't work because you're building like a model on top of a model, so it's gonna be a mess.
But do you foresee that we can get there in the near future?
Define near future.
5 to 10.
Sure, why not? Like, I think we should focus on surviving this year, considering how it started.
Good point.
And then maybe we can talk the next year about predicting ligand binding from sequence. So, a few more points.
Yes.
Does using protein surface information and geometric deep learning provide an advantage of other methods? I would say yes, and I proved it in the paper by comparing it to other state-of-the-art methods.
Right.
So I think we're good with that one. What if you tackle this problem using the amino acid sequence? So I was not sure what you meant with this. So if you want to do it predicting using only the amino acid sequence, then it's going to be very, very difficult, I would say. And it's going to be a bit of a workaround, right? It's going to be more like using sequence features and using homology, right? You take a sequence and you just find like other sequence parts that bind to it that are very, very similar, right? Or if the question was framed in a way, you can go ahead.
So from what my understanding, this amino acid sequence is not as important as the protein surface itself when it comes to binding properties. Is that the case? So if you have molecules hidden in the middle of the protein that are not interacting, do they contribute much to the binding or the protein properties?
Yes.
Okay, so then this is related to the AlphaFold problem, where you are given an amino acid sequence and you first need to predict actually the protein surface.
You can also just try to predict the properties straight from the sequence, right?
But then you'll be using a lot of less useful information, right?
Well, you use the same amount of information, right? You use AlphaFold. You start out with the same information. You just predict model A and then use output of model A to predict by model B, or you just make a model that cuts out the middleman and just predicts B straight away. I wouldn't know which worked better, but like I said, if you want to use only sequence, it's going to be difficult. This is all I'm saying. So the protein structure is already way more informative, and you can see it works. Now it works like good, but if you do the sequence only, it's going to be really difficult. Maybe do slightly better than render.
So which one do you think, when it comes to processing power, requires more processing power?
What do you mean? What requires more processing power?
Using amino acid sequence or processing mesh information, or creating a model that processes mesh information or amino acid sequence?
I would definitely say amino acid sequence.
Would require more processing power?
Yeah.
Yes.
Because if you want to use amino acid sequence, you have to do multiple sequence alignments, right? You need to do sequence alignments and multiple sequence alignments, and they are very expensive to do. It's part of your future generation.
Mm-hmm.
And then you need to do it against like a big database, like a catalog, right? So you want to combine this gene against all similar genes in all other species genomes, right?
Okay, so when it comes to flexibility, which one would be more flexible?
What do you mean by flexibility?
So, like with this, when you're using surface, how much flexibility do you have? That you have one surface like a three-dimensional mesh structure that you're using compared to amino acid sequence. You already have the amino acid sequence, but let's say you don't have the surface information. So in that regard, you're limited with systems where you don't have surface information.
So if you can do a model that performs just as good using only amino acid sequence, it will be considerably more powerful.
Okay.
And more useful. Because the sequence, you can always get the sequence.
But I mean, we know that the surface is the most important feature, right, to have. And any model that's using amino acid sequence implicitly would be learning about the protein surface implicitly. So I think it would be harder to teach it.
It's like you're comparing two problems in a nutshell, right? So one, you put in a protein structure, right? And then it can say, okay, this surface of the protein structure is interacting, right? Look at the second use case, right? If you have a model that can predict the same thing just on the sequence, then you put in a sequence and it's going to tell you, oh, these 5 or 7 amino acids, there are interacting amino acids, right? So you still wouldn't know the surface at the end of it, right? You don't know the 3D structure. You just know which are interacting on the sequence, right? So even the output information would be very different.
So there was a comment about the structure of protein. I just want to point out that proteins don't have a static structure. They're dynamic structures. So they can change. They don't have one structure only. I just wanted to make that comment. And in regards to the surface information, the surface information is a consequence of the amino acid sequence. So you can if you have a model that works well with the sequence, you can basically skip the surface information because you already have that knowledge as a consequence of the amino acid sequence. But will it be easy to create that model? Not as easy as a model that just works with the surface.
I think it's going to be close to impossible. So technically, the amino acid sequence describes the structure, yes. Practically, there are a lot of other factors that helps creating the protein structure. There are like scaffold proteins and many other factors and other things that can affect the protein structure, even when it was folding. So there is a strong correlation between sequence and structure. I'm not gonna deny it, but it's also like there are a lot of complications, right? And people said like proteins exist in a dynamic field. So say you have one sequence, right? And it can have multiple structures, right? And the structure we see from the X-ray crystallography is already the denaturized structure, right? It's already formed. It makes a salt, right? It makes a salt crystal. So you don't really see it might not even be the natural structure of the protein. Not necessarily, right?
Yeah, that's one of my other concerns with work like this, because if you have a dynamic structure, and especially with the ligand binding, if you have a dynamic structure that is going to change, and you're looking at a specific surface structure, the information that you can get from it is not going to be super accurate unless you select a structure that is expected to bind with that structure. So I think the authors kind of talk about this. They talk about creating transformations of this structure, but I'm not sure exactly what they mean. Maybe you can comment about that.
To put it into a nutshell, right? So there are people already, papers out there saying, oh, we found this compound that can bind to COVID-19 as an inhibitor. And this compound is already FDA approved, right? So even if you come out with a compound, a new compound, right, that can kill all the influenzas, right? So you still need to do the human trials, right?
Yeah.
You need to do 3 rounds of human trials. And that's basically the main reason, or one of the main reasons you have to do it, right? Because there's so many things we don't know, right? And there's so many uncertainties. So that's why you still need to double check with the trials, right? You do animal trials, you do human trials. And like there are a lot of like downfalls or problems with not being just approximation of everything, right? Our understanding of the human cell, or our understanding of all these processes are very limited, and everything we have, they serve more or less just as approximations of the ground truth. And some parts of the cell we understand really, really well. Some of the parts are still we don't understand well, and some of the parts we don't even know that we don't know, right? Think of it like known unknowns and unknown unknowns. And so far, this approximation served us really well, right? So these protein structures and this kind of all this research, it did help. It did create a lot of like treatments and other breakthroughs, right?
Yeah, that's right.
So even if it's not perfect, right? It's noisy. Like all this data is super noisy.
Yeah, it's better than not having any tool at your hand. So it's not that they're not useful at all. They're useful, but they're not perfect. Nothing is perfect.
Yeah. Yeah, okay. I guess we talked about the last point as well.
Yeah, we kind of talked about the last point. Okay, let me just check if we have questions online that we want to address. I'm not sure about this. Let's just talk about it. They say by 3D structure. I think I tried to ask this question, but let's just make sure I asked it correctly. They say by 3D structure available, do you mean that the protein has rigid structure at all? Not sure what they exactly mean.
So the protein you consider in this paper, you would consider them as like fixed structure, right?
Yeah. So I think we kind of talked about this. The assumption here is that the structure of the protein is frozen, but that's not the case in real life. So we're just looking at one of the structures of that protein.
Like he is right. There are models and there are methods to deal with this as well. Like there is like simulated annealing, and there are kind of other annealing methods. So if we were talking at the very, very beginning, I said there are like 3 secondary structures, like those alpha helix and those beta folds and the loops, right? So the loops, they're very mobile. They can move a lot and change a lot. Whereas those beta folds and alpha helix, they're like more static. But yeah, everything's in motion, more or less. But say the active site, it's not the motion that's like the big headache. It's like if you have some kind of cofactor, right? So there's an active site, and the active site is inactive. And just the crystal structure you have right now is just the inactive site. So you would say, okay, it doesn't bind this ligand. But however, there's like another active site, a second active site. If there's a ligand bind to it, the first active site changes, and then the protein becomes active. You see this a lot in biology, right? So you have to deal with these as well.
Yeah, and there's more complication because sometimes the protein changes structure based on the ligand. So it's the two factors, ligand and protein, that are dynamic. They're changing and they're affecting each other. So there's so many levels of complication. We're looking at a very simplified form of interaction here, while in real life the interactions are more complicated. So I think we don't have any more questions online. So with that, Florian, I would like to thank you a lot for this presentation. Very informative. Thank you for taking the time to provide us with this informative presentation. And the audience, thank you so much for joining us. And I would like to thank everybody who was involved. With that, I'll say goodbye to you all. Thank you.
Okay, thank you. Bye-bye.
Bye-bye.